Well, good afternoon. So it's a pleasure to be able to share with all of you the progress that we've been making at Alnylam Pharmaceuticals within our ALN-TTR programs for the treatment of a group of diseases called Transthyretin Amyloidosis. And just for starters, at Alnylam, essentially our drug development platform is focused on the use of small interfering RNAs to harness the RNA interference pathway in order to control gene expression to treat human disease. And essentially, this slide shows, really in a nutshell, the RNAi mechanism, and it all begins essentially when a small duplex RNA within the cytoplasm of the cell is loaded onto the so-called RNA-induced silencing complex or RISC complex, at which time the two strands separate, and the so-called antisense strand remains bound to the RISC complex, and then through Watson-Crick base pairing, seeks out its complementary messenger RNA, binds to it. An enzyme called AGO2 within the complex leads to cleavage of the messenger RNA, and that cleavage leads to degradation of the message and then targeted protein suppression. And this is a very, very potent mechanism for controlling gene expression, whereby it's essentially a catalytic mechanism where one molecule of an siRNA loaded onto this complex can seek out and degrade thousands of copies of its cognate messenger RNA. Our goal at Alnylam is to develop synthetic small interfering RNAs against particular genes of interest, genetically validated targets involved in disease pathogenesis. And then really, essentially, the key then is to be able to get that synthetic siRNA to the cell type of interest, into the cytoplasm of interest of that cell, so it can then engage the RISC complex and mediate RNA interference. And really, the greatest progress that has been made from a delivery standpoint over the last 5 years at Alnylam has been delivery to the liver, specifically delivery to hepatocytes. And we've achieved that delivery through several different formulations. These include lipid nanoparticle formulation, shown on the far left, and then our so-called conjugate formulation, where a triantennary N-acetylgalactosamine moiety is attached to the sense strand of the siRNA duplex. And both of these formulations have undergone evolution over time to more potent forms of delivery. The lipid nanoparticle evolved from first generation to second generation, where with the second generation lipid nanoparticle shown here, we're able to achieve substantial suppression of target with doses that are less than 1 milligram per kilogram. And then the conjugate platform has also undergone evolution from our standard template chemistry to what we're calling enhanced stabilization chemistry that Manoharan will be describing in more detail tomorrow, and that's led to about a 10-fold increase in potency. This slide is showing you data with these various formulations, either LNP or conjugate formulations, in animal models against either TTR messenger RNA, the first two panels, or antithrombin messenger RNA. But it's really been this progress in delivery to hepatocytes where we can now have shown in multiple different species, including, most importantly, man, and across dozens of different targets, that we can silence targets within hepatocytes at will using these various formulations, and we can do that safely. And so this progress in delivery to the liver has now really opened up our clinical pipeline. And our clinical pipeline now consists of multiple different drugs for multiple different disease indications. The furthest along are two drugs for transthyretin-mediated amyloidosis, shown at the top. The uppermost called Patisiran, which is our second-generation lipid nanoparticle formulation, which is now in phase 3 in patients with familial amyloidotic polyneuropathy. And then close behind that is ALN-TTRsc, which is our conjugate formulation, also targeting TTR for the cardiac indication for patients with cardiomyopathy. And then you can see closely behind it, we have a number of programs that are based on the enhanced stabilization chemistry conjugate, with ALN-AT3 for hemophilia being in phase 1. We announced this morning some very exciting results showing knockdown with very low doses of this conjugate at doses as low as 0.03 milligram per kilogram. That particular program is in phase 1, as I mentioned. And then you can see coming up behind it multiple different programs that are in the preclinical phase, some of which will be in the clinic next year, including programs in paroxysmal nocturnal hemoglobinuria, targeting complement C5, hepatic porphyria, hypercholesterolemia, and a number of others shown on this slide. So again, the progress in delivery has opened up our clinical pipeline and allowed us to start to advance programs into both early and later stages of development. So coming back then to the programs that are furthest along in our pipeline, which are our transthyretin-mediated amyloidosis programs. So what is this disease? What is transthyretin-mediated amyloidosis? Well, this is a rare orphan disease, and it's essentially caused by a protein that is produced predominantly by the liver called transthyretin. The primary function of this protein is to transport vitamin A, and to a much lesser extent to transport thyroxine. Mutations in this particular protein lead to misfolding of the protein. This is normally a tetrameric protein. The mutations lead to destabilization of the tetramer into the monomeric form, misfolding of the monomers, and then deposition into tissue as amyloid. And depending on the type of mutation, and there are over 100 different mutations that have been defined, one can either develop a predominant neuropathic form of the disease called familial amyloidotic polyneuropathy, or a predominant cardiomyopathic form of the disease called familial amyloidotic cardiomyopathy. And importantly, actually, it's not just the mutant transthyretin that deposits as amyloid, it's also the wild type transthyretin that deposits in the tissues. And the target tissues, of course, are peripheral nerves, gastrointestinal tract, and the heart as well in patients who have cardiomyopathy. And the average life expectancy is approximately 10 to 15 years. The disease progresses inexorably to death in all patients. A significant amount of morbidity and mortality, therefore, associated with the disease. So a high unmet need for new therapeutics. So the siRNA that we have developed for the two programs is targeting the 3' untranslated region of the messenger RNA for TTR. This allows us to essentially capture all different mutant forms of the protein as well as the wild type, again emphasizing that we need to be able to knock down hepatic production of both wild type and mutant protein in order to affect the disease. And again, I didn't mention this earlier, but about 99% of circulating transthyretin is coming from the liver, and it's the circulating transthyretin that is depositing in the nerves, the gut, and the heart and causing disease. So our therapeutic hypothesis for these drugs is fairly simple. The liver is producing mutant and wild-type TTR. The mutation is leading to the circulation of unstable tetramers. These then dissociate into their monomers, which then deposit within tissue, forming fibrils and then amyloid plaques, which then lead to neuropathy as well as cardiomyopathy. So the therapeutic hypothesis is that using either the lipid nanoparticle formulation or the conjugate formulation is to target liver production of transthyretin, both mutant and wild-type transthyretin, and by shutting off the production of transthyretin by the liver, essentially reducing the amount of circulating amyloidogenic protein, thereby preventing further deposition, and hopefully with time, even leading to regression of established amyloid deposits, so that we can see, hopefully, not just stabilization of the disease, but over time, even improvement. Now, what's some of the evidence that lowering transthyretin actually can lead to regression of established TTR deposits? And I'm showing on this slide an experiment in a human V30MET. V30MET is the most common mutation causing FAP. This is a transgenic mouse model with the human V30MET TTR. Within this particular mouse model, these are older mice that already have established TTR protein deposits in the expected target organs, in peripheral nerves and in gastrointestinal tract. And essentially, these mice are given sub-Q administration of a conjugate targeting TTR, so-called siTTRsc, at varying doses. And they're given that for 12 doses on a weekly basis. And over time, we measure their TTR levels and look at the TTR area under the curve in terms of circulating TTR. And then at the end of the experiment, we look at TTR deposition by immunohistochemistry. And you can appreciate the nice correlation within all these different target organs between the level of TTR knockdown. If you look at the absolute TTR concentration, the AUC on the x-axis, as it gets lower with increasing knockdown with higher doses of drug, one sees less in the way of TTR protein deposits. And I think this is a good illustration of our expectation that lowering TTR over a prolonged period of time, lowering serum TTR should and may well result not only in the cessation of further deposition of amyloid, but could also result in the regression of established deposits. So Patisiran is the name of our lipid nanoparticle formulation, formerly called ALN-TTR02. This is the drug we are developing for FAP. Essentially, we have orphan drug status for this drug, both in the US and in the EU, and fast track designation for the drug by the FDA. We have positive phase 1 proof of concept results in human volunteers that we published last August in the New England Journal. We now, importantly, have positive multi-dose phase 2 data in actual FAP patients, and I'll show you some of the highlights of those data. These patients on phase 2 have now rolled over onto an open-label extension study, which allows for more protracted therapy over years that will allow us to now start to look at clinical endpoints in an open-label manner. And most importantly, last December, we started our phase 3 trial, the so-called Apollo phase 3 trial in FAP patients. I wanted to just show this picture quickly. This was our proof of concept with Patisiran in healthy volunteers, published last August. And what I wanted to just really emphasize here is that we're showing that a single dose of intravenously administered Patisiran results in very rapid and potent and durable suppression of circulating serum TTR. And if you look at the various dose groups, and here we're looking at the mean suppression of TTR, percent reduction of TTR relative to baseline, when you get up to dose levels in the 0.15, 0.3, 0.5 milligram per kilogram range, that's where you see greater than 80% suppression, and in fact up to 94, 95% suppression. Again, after just a single dose of drug that persists, with greater than 80% suppression out to past 4 weeks after a single dose. So following this demonstration of single-dose pharmacodynamics in healthy volunteers, it was important for us to establish the pharmacodynamics with multi-dosing in actual FAP patients. And so we did that in a phase 2 study where patients received 2 doses of drug given either 3 weeks apart or 4 weeks apart at the higher doses. So on the left, we're looking at dose response and duration of knockdown by dose group. The highest dose level was 0.3 milligram per kilogram, shown by the turquoise and by the red triangles. The turquoise triangle was dosed every 4 weeks. In the red triangle, every 3 weeks. And what you can see, as I showed you in the healthy volunteers, is this rapid, potent suppression of greater than 80% serum TTR after the first dose. And then that suppression actually persists even right up to the second dose, and then one can see continued maintained suppression. And in fact, when we look at the every 3 week dosing, we can see better, more maintained suppression of more than 80% from baseline with every 3 week dosing versus every 4 week dosing at the top dose of 0.3 milligram per kilogram. On the right, we're able to make another important point here, is that in the patients who had the V30MET mutation, where we had a mass spec assay to look at mutant versus wild-type protein, we see a very nice correlation between knockdown of mutant protein and knockdown of wild-type protein. And that's what we expected, since we're targeting the 3' untranslated region. We expected to see a similar effect on both species of the protein, but it was gratifying to see that in this clinical trial in patients. Now, a number of the patients on this study were also on another drug, which is a small molecule. These are called Tetramer Stabilizers. This is another interesting approach to treating this disease, which is to stabilize the tetramer. These Tetramer Stabilizers bind to the thyroxine binding site within the protein. And the stabilizers that are in clinical development include tafamidis, which is approved in the EU for early-stage FAP, and diflunisal, which is a non-steroidal anti-inflammatory drug, which also has activity in FAP, as shown in a recent phase 3 study. And as you can see on the left, when you look at the TTR levels in the blood in patients who are on stabilizers, as you might expect, you can actually see an increase in TTR levels because you're reducing clearance of TTR from the blood. And in fact, here it looks as though diflunisal at the dose used may be a somewhat more potent TTR stabilizer because the levels go up to a greater extent than with tafamidis at the dose used in these patients. But what was important for us was to show that regardless of whether patients were or were not on tetramer stabilizers, that we saw the exact same pharmacodynamic activity with our drug, and that's shown on the right side, where patients, whether they were on diflunisal or tafamidis or on no stabilizer, if we look at TTR knockdown after the first dose at 0.3 milligram per kilogram, we're seeing the exact same impact. And then lastly, I also wanted to show some of the PK/PD that we were able to analyze in the study as well. And we saw a nice correlation between AUC of plasma siRNA and the amount of knockdown, percent knockdown of TTR in the serum. And if you could look at the various colored bubbles, looking at the various dose levels, you can see this dose response effect on TTR knockdown and this corresponding correlation between the AUC in the first 24 hours for the siRNA level in the plasma and the degree of knockdown that we see. And then in terms of safety, we saw that the drug was very well tolerated. If you look at the various treatment-emergent adverse events, there were few of them. Most of them were mild or moderate. And if you look across the table at the higher doses, 0.3 milligram per kilogram every 4 weeks or every 3 weeks, the only adverse event that we really saw with the every 4-week dosing of any significance were infusion-related reactions, but these were predominantly mild, self-limiting, and dealt with usually just by slowing the infusion. None of the patients had to discontinue because of infusion-related reactions. We saw several serious adverse events. One was from extravasation of drug that was managed just with local care, and the other was a patient who had gastrointestinal symptoms who also had pre-existing gastrointestinal symptoms from autonomic disease from his FAP. And importantly, no changes in liver function tests, renal function, or hematologic parameters. So very well tolerated at these doses and with the every 4-week or every 3-week schedule. So as I mentioned earlier, we've moved on to an open-label extension study. Patients on the phase 2 study were all eligible to roll over onto the extension study, which allows up to 2 years of dosing with clinical endpoints evaluated every 6 months. And these include endpoints, key neurologic endpoints that are all on our Apollo phase 3 study. The regimen is 0.3 milligram per kilogram every 3 weeks. And for patients who are doing well at 2 years, they can roll over onto another extension study. We also have a subgroup of patients where we are looking at serial echoes and cardiac biomarkers for those patients who have cardiac involvement when they roll onto the extension study. The study objectives are shown here. Again, I would focus on the secondary objectives with regard to the sorts of endpoints that we're looking at here, the clinical endpoints, which will give us important readouts every 6 months that parallel the sorts of endpoints that are on our phase 3 study. We have 23 patients now with data that we can analyze and share. We have 25 patients enrolled to date, and we expect to have 27 out of the 29 patients roll over onto this study by the middle of this month. So looking at the demographics for the open-label extension study, for the 23 patients for whom we have data that are shown on this table, we had a variety of different TTR genotypes. The most common was the Val30 MET, but we also had other so-called non-V30 MET mutations. Most of these patients had relatively early stage disease. A good number of the patients, the majority of patients, were on a concurrent Tetramer stabilizer. And at the time that this was presented initially in early April, patients had received anywhere from 1 to 8 doses on this study. So this is really our first experience with more than 2 doses with this drug in this patient population. And so far, the safety profile has been very encouraging. All of the treatment-emergent adverse events, and you can see there are very few listed here as being related or possibly related, were mild to moderate in severity. We haven't seen any serious adverse events, no changes in liver function, renal function, or hematologic parameters. And we've only seen an infusion-related reaction in one subject. That one subject had them after the second and the third dose, but these were very mild and did not even require any slowing of the infusion or any interruption of the infusion. Now, interestingly, what we've explored both in phase 2 and now in the phase 2 extension study is an alternate way of infusing the lipid nanoparticle. As I mentioned, one of the side effects one can see with these lipid nanoparticles, albeit at a low rate, given the low doses we're using, are these infusion-related reactions. And we actually did some interesting experiments in pigs to show that one could mitigate these reactions by giving the lipid nanoparticle slowly at first and then speeding up the infusion rate. So starting with the phase 2 study and then on the phase 2 open-label extension study, we migrated from using a 60-minute infusion, where the drug is given at 3 minutes per mil for the entire 60 minutes, to giving what we call a microdosing regimen, where for the first 15 minutes we drop the rate to 1 mil per minute, and then afterwards we go back up to 3 mils per minute and finish the infusion. So it ends up being a 70-minute infusion instead of 60 minutes. And if you look at the doses associated, percent doses associated with an infusion-related reaction with the 70-minute infusion compared to the 60-minute infusion, you can see a significant reduction in the incidence. And so this is something that we have been very encouraged to see in the open-label extension study, and this 70-minute microdosing regimen is what we're also moving forward with in our Apollo phase 3 study. And then importantly, I think one of the important questions is of course, well, with long-term dosing in the extension study, are we maintaining suppression of transthyretin? And here, these are preliminary data where you can see initially we have data pre-dose and then after the first dose. And after the first dose, we have data through day 17 and 19 of the patients. Beyond the first dose, we have so far limited amounts of data, either right before the third dose, right before the fifth dose, or right before the ninth dose. So these are pre-dose. So this is a conservative estimate of knockdown, that this is probably the least amount of knockdown that you would see because you're seeing it right before the next dose. But you can see that even looking at it this way, we're able to maintain, at the very least, 80% suppression of transthyretin, even at these pre-dose draws, which is very encouraging to us to know that even at the troughs we're getting recovery that's no more than 80% knockdown, and being able to maintain TTR suppression of 80% from baseline or even more, we think is important for fulfilling the therapeutic hypothesis behind the drug. So this is very encouraging efficacy data so far from a pharmacodynamic standpoint. So to give you a brief summary of the phase 3 trial in FAP, the so-called Apollo study, this is projected to be a 200 subject study with 2-to-1 randomization. We're looking at patients with all different mutations, V30-MET and non-V30-MET. We're looking at a wide range of neuropathy severity with an NIS score of 10 to 100. And we're going to be looking at 18 months of treatment with the 0.3 milligram per kilogram scheduled every 3 weeks versus placebo. And the primary endpoint is a composite neurologic impairment endpoint called the modified NIS plus 7. And then we have a number of secondary endpoints that are also measuring how patients feel and function, including quality of life, disability scores, timed 10-meter walk, nutritional status with modified body mass index, et cetera. And so we think we will learn a lot from this study with regard to its effect on neuropathy and also with regard to its effect on quality of life and the overall condition of these patients. So then turning finally to the second program, the ALN-TTRsc. This is being developed for the cardiomyopathic form of transthyretin amyloidosis, the so-called familial amyloidotic cardiomyopathy. This is given subcutaneously as opposed to through intravenous infusion. We now have positive phase 1 study results in normal healthy volunteers, and I'll show you those data in a moment. We're in the midst of a phase 2 study in FAC patients, and we plan on starting phase 3 in FAC patients by the end of 2014. So these are the phase 1 study results in healthy volunteers, and here we're using a regimen where we're giving subcutaneous injections of ALN-TTRsc once a day for 5 days, and then weekly for 5 weeks. So a total of 10 doses over 5 weeks, and you can see that the duration of the dosing goes out to where that purple bar is along the x-axis. And we looked at placebo as well as 3 dose levels, 2.5, 5, and 10 milligram per kilogram. And you can see the very nice dose response with greater than 80% and actually even greater than 90% suppression at dose levels of 5 and 10 milligram per kilogram. And you can see that that suppression is sustained even when dosing is discontinued, and then recovery gradually occurs within the next 30 days or so afterwards. What's always been very encouraging to us, both with the lipid nanoparticle formulation and with the conjugate, is the striking correlation between pharmacology in the non-human primate as well as in man. And on the right, we're giving an example of that, where if we look at a single 10 milligram per kilogram injection of ALN-TTRsc, either in the monkey or group of monkeys or in the cohort of healthy volunteers, you can see a similar nadir. But actually, you can see one interesting difference is that it looks as though the duration of the effect is somewhat longer in the human subjects as opposed to the non-human primates, suggesting that there may be a different nuclease environment that may be somewhat attenuated in man compared to the non-human primate, giving us a longer duration of action. From a safety and tolerability standpoint, the drug has been very well tolerated so far. All of the treatment-emergent adverse events have been mild or moderate in severity. We've had no serious adverse events. The most common sort of adverse event has been mild injection site reactions that appear to be somewhat dose-dependent. These typically consist of limited erythema that's usually asymptomatic, lasts for several hours, and then resolves. Again, we've had no study discontinuations, no flu-like symptoms, even at the highest doses, and no lab abnormalities. So we are now in the midst of our phase 2 study. This is an open-label, multi-dose study in patients with the hereditary form of cardiomyopathy called FAC, and also in patients who have the wild-type form of the disease called senile systemic amyloidosis. We plan on enrolling up to 25 patients on this study using the 5 milligram per kilogram dose. And again, it's a 10-dose regimen, the 5 daily doses followed by 5 weekly, assessing safety and tolerability, but also we have a number of important pharmacodynamic endpoints looking at knockdown of total TTR as well as wild type versus mutant protein, and a number of exploratory clinical endpoints within the study, including cardiac imaging, 6-minute walk, et cetera. We are planning to open an open-label extension study in the middle of this year, so patients will be able to roll over onto this, from this study onto an open-label extension study and receive dosing for up to 2 years with these clinical endpoints that will be followed. So in summary, for these 2 programs that are being developed for the treatment of transthyretin amyloidosis, Patisiran showing very robust and durable knockdown in patients with a multi-dose regimen. This phase 2 open-label extension study ongoing has been showing very encouraging pharmacologic activity as well as safety. And we expect in late 2014 to have our first interim 6-month look at clinical endpoint data that we'll plan on sharing at a major meeting at that time. And of course, our Apollo phase 3 study in FAP is ongoing. And in terms of our program for FAC, the ALN-TTRsc program, we have the positive proof of concept data in healthy volunteers, the ongoing study in patients with TTR cardiac amyloidosis, and are moving now rapidly toward initiating a phase 3 trial within this population by the end of 2014. And lastly, I just want to acknowledge all the key investigators, the phase 2 investigators, the so-called Patisiran investigators on the left, and then our 2 key phase 1 investigators, as well as some of our important scientific collaborators over the years. Thank you very much.